Good day to all. Let us now start knowing about the basic tissues of the body. So what is cell? Cell is the basic structural and functional unit of all the tissues in the body. And amazingly, as we said that a single cell, the fertilized ovum, which is the zygote, gives rise to hundreds of different kinds of cells, which compose to about 100 trillion cells that make up the adult human body totally. So then the cells, group of cells performing the similar function, they form a tissue. So there are about 4 basic tissues which are the building blocks of the human body. They are the epithelial tissue, which covers the surface of the body and the hollow cavities. And then the connective tissue. It joins and supports other tissues. Then the nervous tissue. It conducts impulses from outside and inside the body. The muscle tissue. By contracting this tissue, moves the parts of the body and also moves the solids, fluids and air along the GIT, CVS, and that is the cardiovascular system and the respiratory system. The movement of the muscle which is causing the movement of these structures is called as peristalsis. We'll come to know about all these things as we progress in our lessons. And all these 4 basic tissues constitute to form an organ. And similar functioning organs, they form an organ system, and different organ systems will form a complete human being. Epithelium is a sheet of cells that covers the body, lines the inner and outer surfaces of the organs, lines the body cavities, forms the glands, and lines the hollow structures, the ducts, all these things. Any hollow structure in the body is lined by epithelium. And the cells are tightly packed with minimal intercellular substance. So they are bounded by junctional complexes. We'll come to know about them later. And they lie on the basement membrane, separating the epithelium from underlying connective tissue. The epithelium is avascular, so there is no blood supply to the epithelium. So the nutrition occurs by mere diffusion, but it is nervous, that is nerve supply is there. Epithelium has a very good capacity of regeneration, where it undergoes mitosis and replaces the damaged cells. For example, you got hurt, there is an abrasion of the skin, and over a period of time, probably a week's time, you will see that the skin has been regrown. You won't be able to differentiate that area from the other area where you might have got injured. So that's how the epithelium, it undergoes mitosis and regenerates the damaged area. It shows polarity. It shows surface modifications like the cilia, the villi, the microvilli, the gastric pits. So these are all the surface modifications where the epithelium has. And the most important thing is it shows metaplasia, which is an abnormal side of it, where the cells change from one type to the other. Like, for example, a common thing I'll tell you. A person who smokes, who is a chain smoker, The respiratory epithelium, which is a pseudo-stratified ciliated columnar epithelium, because of this pressure and the stress that occurs on the epithelium with these carbon particles which enter through the smoke, that epithelium will be converted into a stratified epithelium. That is the reason they lose cilia. So the basic function of the respiratory epithelium is lost. So that is the reason the smokers, when the chain smokers, we may say that, they cough so much and end up in various other respiratory complications and lung diseases. Okay. And then the most important thing to know is the epithelium is derived from 3 germ layers. So this is the germ disc. The blue one, what you are seeing, is the ectoderm. The yellow single line lower is the endoderm. In between, the red color is the mesoderm. So there are various organs that are developing from the ectoderm, mesoderm and endoderm. So as in when the organs develop, the epithelium which is lining those or covering those organs also develop from the same germ layers. So from ectoderm, the skin is developed, that is the epidermis, glandular tissue is developed, the glands, breast is developed and cornea is developed. From the endoderm, it is the lining of gastrointestinal tract and respiratory tract are developed because these two organs are developed from endoderm. And the mesoderm gives rise to lining of the urinary system, the cardiovascular system, the peritoneum, which is lining the body cavities, and the pleura. So the cell has 3 surfaces. The basal surface, which is resting on the basement membrane. In the lateral surface, that is on the sides, which is in contact with the adjacent cells, and the apical surface, which is the surface of the epithelial cell. Now coming to the functions. The very important function of epithelium is protection. It protects the deeper structures. For example, skin. The skin is protecting the deeper structures, the muscle, the vessels and all that. So that's how the protection is done. And it also acts as barrier. It acts as a mechanical barrier and permit selective substances to cross it. For example, again, the epidermis, it prevents the entry of viruses. So when we go out in the air, we have so many other disease-causing organisms and all. That's how it is acting as a selective barrier by preventing the entry of certain organisms. Absorption, it is again a selective function where, again, for example, in intestine, epithelium absorbs the nutrients from the digestive food and then undergoes various metabolisms and then secretion. It can synthesize and secrete some products. For example, salivary glands, they secrete saliva, and the gallbladder, the cells of the gallbladder, they secrete bile and things like that. And the sensory perception. It is involved in sensory signals which they take from the external environment, like for example, tongue. A person can perceive the taste from various food particles that touch the tongue epithelium, and hearing, internal ear. By the sound waves, the sensory perception is perceived and the person hears. So that is what is about basic features and the structure and function of the epithelium. There is one thing to know, as there are few cells which are called as epithelioid cells, which are not epithelial cells. They are immune responsive cells. Okay, so they are called as modified and activated macrophages, which resemble epithelial cells, which are elongated with pale eosinophilic cytoplasm, that is pink color one, with usually an elongated nuclei, and they often merge to form a giant cell. So these pale looking cells are all epithelioid cells. Okay, and when they merge 4 or 5 cells, suppose, they become into one cell with 4 or 5 nuclei. So they become giant cells which have multinucleated. They are called as Langerhans giant cells. And these epithelioid cells are very much a feature of granulomas. These granulomas are nothing but they are formed due to chronic inflammation. Okay, so when a person usually seen in tuberculosis, so this is what the lymph node of a tuberculous patient is taken out. So this is all is the necrotic caseated area on the lymph node. It is a very much enlarged lymph node, infected lymph node. Okay, so when an area of the lymph node is infected and dead, that becomes into a cheesy material which is called as caseous material, that is called as caseous necrosis. So when such type of thing is there, it should not extend to the whole of the lymph node. So then these epithelioid cells, they surround this caseous material. This is the caseous material. So these epithelioid cells, they surround the caseous material, preventing the spread of this infection to the further areas. And this these epithelioid cells are again surrounded by lymphocytes, which are again immune responsive. So that's how these resemble epithelial cells in a way that they also line or surround the caseous necrotic material directly on the surface of it, forming a boundary zone of delimitation, so that it does not, the pathogen does not spread from this area to various other areas on the lymph node. So there are various places these granulomas are formed in this. So this is a tuberculous lymph node. So this is one thing which has to be known, where the epithelioid cells are not epithelial cells. They are single cells which are not present on a sheet of basement membrane. They are scattered. They are present surrounding, lining the caseous necrotic material, which is called as granulomas, preventing its spread. So what is basement membrane? It is a thin sheet of extracellular matrix secreted by epithelial cells which are resting on it. So this black line, what you are able to see, the dark line, is the basement membrane. These are all the epithelial cells. They are sitting on this basement membrane. Okay, and this basement membrane is secreted both by the cells and by the connective tissue. And this basement membrane can only be seen through electron microscope. What are the functions of this basement membrane? Again, it supports the overlying epithelium, anchors the epithelium to the connective tissue below. It also acts as selective barrier and helps in regeneration of these epithelial cells after the injury. This basement membrane, this is the electron microscopic view of the basement membrane. Okay, this is the cell from here. So this from here till here is the basement membrane. So this basement membrane has two layers. One is basal lamina, the other one is reticular lamina. So this basal lamina is nearer to the cell base or the cell surface, and this reticular lamina is nearer to the connective tissue. And these mainly the basement membrane is made up of collagen fibers, various types of collagen fibers, of which 4, 7 and 3 are of most importance. So this basal lamina, which is divided into the basal lamina as well as the reticular lamina, is composed of type 4 collagen fibers. It has glycoproteins and also the proteoglycans. The basal lamina is further divided into lamina lucida and lamina densa. So the lamina lucida means it is a clear zone adjacent to the cell surface. It is mostly made up of amorphous matrix and integrins are present. The glycoproteins, which are integrins, are present. The lamina densa is a very dense zone between the lamina lucida and the reticular lamina. And this lamina densa has abundantly having the laminin and the 4 collagen, type 4 collagen is present. Now coming to the reticular lamina. The reticular lamina has various reticular fibers, as the name goes, and these fibers are produced by the cells of connective tissue. Whenever we say fibers, they are produced from fibroblasts. So from the fibroblasts, these reticular fibers are produced and they are scattered all over, and this basal lamina and the reticular lamina are anchored to each other by anchoring fibrils. Similarly, the cell and the basement membrane has to be anchored as well by anchoring fibrils. And these anchoring fibrils have type 7 collagen. And these reticular fibers, they have type 3 collagen. And these anchoring plaques within the reticular lamina, they have type 4 collagen. So for example, let me say that this anchoring filaments between the cell and the basement membrane are made up of what we said, type 7 collagen. So when this type 7 collagen is not formed properly, which is a genetic disorder, then what happens is the cell loses its support from the basement membrane. So the cell surface, it gets raised from the basement membrane. and there's a gap that is formed between the basement membrane and the cell, and that gets filled up with intercellular fluid. No space in the body is empty. If there is a space, it gets filled up with the intercellular fluid. So that's how the blisters are formed. Okay, we'll see them later. So this is the summary of basement membrane, because basement membrane is a short note for you. It has two layers, the basal lamina, reticular lamina. Basal lamina in turn has two layers, lamina lucida and lamina densa. Lamina lucida doesn't have or doesn't show any features, but consists of very fine type 4 collagen and the amorphous matrix. The lamina densa has the laminin, the proteoglycans and the type 4 collagen fibers. The reticular lamina, which lies deeper to the lamina densa, between the lamina densa and the connective tissue, they contain reticular fibers which are having type 3 collagen and anchoring fibers which are anchored to the above layers which are having type 7 collagen. And the main function of the basement membrane is supporting, anchoring the epithelium to the connective tissue and to the basement membrane. It acts as a selective barrier. It has a very good regenerating capacity after injury to the tissue. So coming to the surface specializations of the cell. So the only surface which is left free is the apical surface. So on the apical surface, we find cilia, microvilli, stereocilia and flagella as modifications or the specializations on the apical surface. And on lateral and basal surfaces, which are adjacent to some other structures, that is, the lateral surface, it is adjacent to the adjacent cell, and the basal surface is adjacent to the connective tissue, they have junctional complexes. Cilia. So this is cilia. It's an elongated hair-like motile evagination from cell surface. You can see this is the surface epithelium. It has evaginated like this. Okay. So the evagination from the cell surface, which is elongated and hair-like, forms cilia. And this cilia, as itself it is thin and thread-like, it also has slender thread of cytoplasm into it, because it is the evagination of the cell surface. As the cell has cytoplasm, the cytoplasm also enters into the cilia, and the cell surface which is covering the cilia is called as plasma membrane, as it is known, covering the cell itself. So when you look into the cut section of cilia at various levels, that is at the base, in the middle and at the tip, basically the cilia, they arise from the centriole of the cells. They are modified centrioles. So as the centriole has the microtubules, even the cilia has the organization of microtubules in a different way at various levels. So at the base, the microtubules, these are the microtubule, they are arranged in triplet fashion. There are about 9 peripheral tubules. They are arranged in a triplet fashion. And When you see the cut section at the middle of the cilia somewhere here, it consists of the central pair of microtubules, and on the periphery it has 9 pairs of microtubules which are arranged in pairs, that is twin filaments. Okay, so there is a central microtubule, double pair of central microtubules, and on the periphery there are pair of 9 microtubules that are present, and each microtubules, they are connected by the nexin, which is a protein, this one, and from the periphery the microtubules are connected to the central microtubule by radiating spokes. The peripheral 9 microtubules, the twin filaments, they have a very important protein which is called as dynein. This dynein is the main protein that is responsible for the motility of the cilia. The main function of presence of cilia is to have motility for that concerned area. Okay, so like in respiratory tract, like in uterine tube, like in ependyma, like in olfactory epithelium. So respiratory tract, it has to push out the foreign particles, the dust what we inhale, which settle on the respiratory epithelium, has to be sent out, otherwise it goes inside and infects the lungs. The uterine tube, it has cilia because it has to propel the egg to get fertilized with the ovum, and after the fertilization that zygote has to be transported into the uterine cavity for it to get implanted. Ependyma, which is present in the ventricles, it has to move to circulate the CSF. Olfactory epithelium, which is on the roof of the nasal cavity, also has almost the same function, where it is sensory. It takes in the receptors where we inhale the air, it senses the smell. Smell sensations are taken by these olfactory epithelium, which has the cilia. So all this movement of the cilia is mainly because of the protein dynein. If there is any damage to the formation of dynein, if the dynein protein is not formed properly, the motility of the cilia is severely affected, which will hamper all these functions what we said. So this cilia, which is present in the respiratory tract, it is called, the function of it is called as mucociliary escalator. All of you know the escalator, right? In prasads and various other theaters, the multiplexes, it has escalator where you just stand on it and it goes up, taking you to the upper floor. So the dust and all which is settling in the lower respiratory tract also should move up the trachea and to the nasal cavity to get it out on the mucus. So the movement of cilia should be in that direction, only in the upward direction. So that function of cilia in the respiratory passage is called as mucociliary escalator, where cilia is propelling the mucus containing trapped particles towards the pharynx. And then this cilia is never present on any of the stratified epithelium. It is only present on the simple epithelium where the epithelium rests on the basement membrane. Okay. And when you take, this is the section what you see in the middle of the cilia. So when you go on to the tip of the cilia, when you take the section, you just find only the central microtubules without the surrounding 9 microtubules. So that's how it becomes tapering towards the apex of the cilia. So at the base it has triplets, in the middle it has twin filaments, and in the tip of the cilia it just has the center pair of microtubules only. And this condition where the Dianin function is not proper, it is called as Cilia Syndrome, where it is a hereditary defect in ciliary Dianin. The lung infection will be there as cells fail to clear the debris and bacteria which is present in the mucus of the respiratory tract. And wherever the cilia is there, right, the cilia function is not proper. So in the uterine tube also, the males and females, they are sterile, which is called as immobile ciliary syndrome or immobile cilia syndrome. And the cilia of ependyma also doesn't function properly, so there is accumulation of the CSF which can lead to hydrocephalus, and the loss of sense can also be felt because of the loss of respiratory epithelium. The cilia present on the left respiratory epithelium also doesn't function properly. So this is what is a picture for cartilaginous syndrome, which is also called as ciliary dyskinesia, where the cilia doesn't move. So this is the normal respiratory epithelium which has cilia, and all are moving in one direction like this, so that the mucus which is settled over the epithelium move upwards to throw off the bacteria and dust if they're into the pharynx. But if the Dianin is not properly formed, when there is a defect in the formation of the protein Dianin, then there is irregular movement or no movement for the cilia. They're just present. So the mucus which is there on the respiratory epithelium cannot be moved above. It can't be escalated through the respiratory passage to the pharynx. So it gets piled up here and it may go down into the lower respiratory tract, infecting the lungs. Along with this respiratory abnormality, there will be infertility problems in males and females because of the cilia being not functioning in the uterine tube. There can be hydrocephalus. All these are associated symptoms of this immobile Cilia syndrome. Now coming to the microvilli, another modification of the apical surface. These are small finger-like extensions from the cell surface, and they are covered by glycocalyx. They are not tapering structures, they are not long structures, they are just small finger-shaped little stout structures. These are present mainly to increase the surface area for absorption. So within this core of microvilli, there are about 20 to 30 actin filaments, these longitudinal structures. They are connected to the horizontal actin filaments of the cell. And these vertical actin filaments are connected to one another by the protein vilin and fibrin. Okay. And these actin filaments are connected to the cell surface by myosin. Since these actin filaments are connected to one another, there is very minimal mobility or no mobility, we can say. And at the base, this is the base, right? There is a terminal web, which is nothing but the horizontal meshwork of actin filaments, which are connected to the vertical filaments which are present in the microvilli, which will confer a very less or no movement to the microvilli. And these microvilli are arranged in two types. If they are regularly arranged, they are called striated border, which is a common feature in small intestine. If they are arranged in an irregular way, not to the same height, one are tall, one are less in its height, things like that, when they are irregular, they are found in gallbladder and proximal convoluted tubule of the nephron. And if there is no proper actin filament formation, that leads to loss of absorption. All metabolic diseases the person will end up in, because the main function of microvilli is the absorption. The microvilli is present only to increase the surface area of absorption, which is taken care by the actin filaments which are within the core of the microvilli. Coming to stereocilia, these are also like microvilli, little less thicker than microvilli. They are non-motile. They also increase the surface area of the cell. So they are present in epididymis, again for absorption. They are present in cochlea for signal generation. They catch up the waves in the cochlea. And then they are also present in nose, ear, taste buds, hair cells, sensory. They are more into the sensory sensations also. And then coming to the flagella, this long tail like structure, it is the flagella. It is like cilium but much longer. It has the configuration just like the middle part of the cilium, where it has a pair of microtubules in the center and 9 microtubules paired up onto the periphery. And the flagellum is seen only for spermatozoa. Nowhere else in the body, any cell doesn't have the flagellum. The only structure with flagellum is the spermatozoa, which in its future course loses it also.